Welcome to ShepNotes' first presentation of genetics and heredity. We'll begin with a basic introduction. Heredity is the study of how parents pass off traits to their offspring. The study of heredity is often called genetics. It seeks to explain questions like why a group of children from the same parents can look like their parents and still have differences between each other. The phenotype of an individual is the observable characteristics of the individual. These are called traits and include visible properties like hair color, behaviors like being friendly, and how healthy they are. All of these traits are determined by chemical instructions called genes, and the genome is the collection of all of the genes possessed by an organism. The genotype is the inherited genes for a specific characteristic like eye color. Variations of the genes for each trait, like those that determine whether the eye color is brown or blue, are called alleles. One of the alleles is usually dominant over the recessive allele, and it masks the effects of the recessive allele. The basic building block of every living thing is the cell. Specialized types of cells form bones, muscles, connective tissue, sex cells, and everything else required to create organisms capable of growing, reproducing, moving, and interacting with their environment. Most of life's forms that we see and interact with, like other humans, dogs, and cats, are called eukaryotes, and they have cells that have a membrane-bound nucleus. Most of the genes of eukaryotes are packaged in long DNA strings called chromosomes that are located inside the nucleus. Other life forms we have all around us but seldom see, like bacteria, do not have a nucleus in their cells, and they are called prokaryotes. The basic chemistry and gene structure, or genotype, is very similar in every form of life that is found on Earth. This drawing is a simplified view of a DNA chromosome. The ladder-like structure is a close-up of one of the thousands of genes found in a typical chromosome. The rails of this ladder are composed of sugar-phosphate bonds that serve as the backbone of the DNA string. Between each sugar-phosphate bond, the rails are bonded together with one of two base pairs, adenine thymine, labeled as AT or TA, or guanine and cytosine, labeled as GC or CG. Adenine is always bonded with thymine, and guanine is always bonded with cytosine. If the rungs of the ladder get separated, a missing rung can be created by adding the bases matching those on the rung being copied to a new rung. The arrangement of the bases on the DNA string are coded and used as the information used for creating proteins and controlling traits. When a protein is needed by the cell, the portion of the chromosome containing the gene for the specific protein is exposed and one of the rungs is copied. The original base, thymine T, from the DNA is replaced with uracil U. The resulting string, shown in the drawing, is now called messenger mRNA. The mRNA is moved to the ribosomes outside of the nucleus, where the information in the codons described below are used to add the required amino acids necessary to a polypeptide string that will become the requested protein. The codons are groups of three bases, G, A, C, and U. Since there are 4 bases in groups of 3, there are 64 possible codes in a codon. In the example, the codon GGA is the code for the amino acid glycine, and the GUC codon is for valine, which is another amino acid. There are 20 possible amino acids that can be used for a protein. Some amino acids have from 2 to 6 codes to minimize the effects if a base is modified by an unintended mutation. Now let's take a look at Mendel's heredity. Gregor Mendel crossed a tall pea plant with a dwarf pea plant, and these parents were called the P or parent generation. He called the offspring the F1 or first filial generation. In the F1 generation, the offspring were all tall. When 2 of the F1 generation of tall plants were crossed to produce the F2 or second filial generation, 3/4 of the offspring were tall and 1/4 were short. If the parents in the P generation were both tall, or the parents were both dwarfs, the offspring would always be the same height as the parents, and this would be true for each of the following generations, without the alternate trait ever appearing. After his experiments with pea plants, Mendel concluded that some heredity factor must be passed from the parents to future generations to account for the results. Each parent must be able to carry either of any variations of the factor, and each member of the offspring must inherit random combinations of any variation of this factor. Mendel's factors are called genes by modern geneticists. In this example, for the size gene, there are 2 variations, which are called alleles for the trait. The allele for the tall plant is labeled D with a capital letter because it is called the dominant allele. The allele for the dwarf plant, called the recessive allele, is labeled D with a small case letter. When the dominant allele is present in a gene pair, the recessive gene is ignored. This gene pair is called a homozygous dominant allele because it contains the same dominant gene from each parent, capital D and capital D. This gene pair is called a homozygous recessive allele because it contains 2 of the same recessive genes, small letter d and small letter d. These gene pairs are all called heterozygous alleles because they contain both a dominant and a recessive allele, a capital D and a small letter d. The F1 generation are all called carriers since they are all tall plants but carry a recessive dwarf gene. This gene can be passed on to later generations. Their dominant D gene for tall plants masks the effect of the recessive D gene for the F1 generation. This chart illustrates why brothers and sisters and cousins should never marry. If the recessive D allele in this example were a serious disease instead of a plant size, and the chart was for people instead of plants, the parent in the F1 generation would have passed the disease on to their last child in the F2 generation, and they probably didn't even realize they were carriers. Hi friends, this concludes the first ShepNotes discussion on genetics and heredity. Watch for the next ShepNotes edition, which will provide more information and intuition on genetics and heredity.